Okay, this is just a summary of the VCE Biology Unit 3 Outcome 1 area of photosynthesis. So just looking through the purpose of photosynthesis, chloroplasts, so their structure and also their origins from bacteria, the inputs and outputs of each of the stages of photosynthesis, and the factors that affect the rate of photosynthesis. So we start with our equations. With photosynthesis, the goal is to produce glucose. Okay, that is the primary output that we're concerned with. So we have both of our word and chemical equations here. You do need to know both. Always easier to start with the word equations. So we have carbon dioxide and water are inputs. When we write this part of the equation, you need to include these words. Okay, so above the arrow, we write light, which is our energy source, and chlorophyll, which is the pigment that absorbs that energy. So even though they're not molecular inputs, we include them because they're crucial. And then our outputs are glucose and oxygen. And if we go down to our chemical equations, we just put some numbers to those. So it's 6 carbon dioxide and 6 water inputs. We still write light or chlorophyll, and then we have our glucose molecule, so C6H12O6, and 6 oxygen output. It's also important to know that photosynthesis is an anabolic process, so it builds, and in particular it's building a glucose molecule, and it is endogenic, it uses energy. Okay, the process of building something up requires the use of energy. Okay, you should know how to draw a chloroplast. And you definitely need to be able to interpret a diagram of a chloroplast. This often comes up in the exam. So we have an inner membrane on the chloroplast here, as well as an outer membrane. So it's actually, if we were to cut a slice, kind of looks a little bit like that. We then have these membrane sacs here, and they're called thylakoids. Okay, so they are the individual discs that are found in the chloroplast. And then a stack of thylakoids is called a granum, or the plural of that is grana. So this is one granum. And in this chloroplast, I can see 1, 2, 3, 4, 5 grana. The lumen is just the opening inside the thylakoid. And you also need to know that the area inside the chloroplast, which is kind of full of a jelly-like fluid, is called the stroma. Okay, so that is the fluid inside. You need to know all of these things because you need to know where different stages occur. So the thylakoid, these things here, is where the light-dependent stage of photosynthesis occurs. And then the stroma, so the fluid in here, is where the light-independent stage occurs. Okay, you do need to know that chloroplasts are believed to have originated from a type of cyanobacteria. So they evolved from actually being a prokaryote themselves. And they formed an endosymbiotic relationship with a larger host cell. So basically what this means is, a really long time ago, chloroplasts were their own structure. They were actually a type of bacteria. And then at some point, they were engulfed by a larger eukaryotic cell. Okay, until eventually we now have the eukaryotic cell with the chloroplast, which was originally a prokaryote, inside it. And it's an endosymbiotic relationship because they benefit each other. The prokaryote is protected by the cell around it, and now the cell around it is able to produce its own energy source. And these were the early plant cells. Okay, so they would have evolved then to become the more complex plant cells that we know today. The evidence we have that this occurred are the size of chloroplasts. So chloroplasts are quite large organelles, similar in size to small bacteria. They have their two membranes. Okay, so remember, bacterial cells, prokaryotes, have a cell wall and a cell membrane, and chloroplasts have that as well. And they also have their own DNA and ribosomes in them, where most organelles don't have that. Okay, so this is a closer look here at the structure of a chloroplast, which is quite similar in many ways to bacteria. So that is the evidence that chloroplasts evolved from bacteria. Okay, chlorophyll. Chlorophyll is the pigment found in the thylakoids that absorb the light energy from the sun. Remember that light is an important part of our equation, as well as this term chlorophyll. So when light shines down onto a leaf, certain colours of the light spectrum are absorbed better. So you can see up here, this is showing the amount of absorption. Up here, the blue and the purple lights, and over here, the red light are absorbed best. The pigment, the light spectrum that's not absorbed very well are the green and the yellow. And that's precisely why most plants look green or yellow to us, because when light, when the green and yellow light is reflected off that plant, it's not absorbed, that is what we see. Okay, so blue, purple, red are absorbed best, and plants are green because the green light is reflected. It's not absorbed well. Questions have come up in the exam before that would ask what colour light is absorbed best by chlorophyll. All right, just do a quick little summary of some of the things you would have looked at last year, but that are important with this when we think about what is required for photosynthesis. Okay, so water is absorbed by roots and will travel up the plant to the leaves. Carbon dioxide diffuses through these things called stomata, which are found in the bottom of the leaf. Okay, so carbon dioxide from the atmosphere diffuses through there, and then we get the light from the sun comes down onto the tops of the leaves. And this is why most leaves are wide and flat, so they can absorb as much sunlight as possible. Okay, so let's imagine now we cut this little bit of a leaf and we want to look at its structure. So this would be the top of the leaf, this would be the bottom of the leaf. So you can see these stomata here where the carbon dioxide would enter. We'd have our light entering down here, and then there's all these individual cells along there, and you can see these little green dots are the chloroplasts. So if we now went from one of those cells to what we know of a plant cell, we have our nucleus, we have our, that's our vacuole, we have our nucleus, we have our Golgi, and we have these chloroplasts. Okay, so when we're looking at photosynthesis now, we are purely looking at what is going on in one of these things, but it helps to have that bigger picture. Okay, we looked at coenzymes at the end of the last topic, but it's important to recap that here because they play a really important role in photosynthesis. So coenzymes are molecules that bind to an enzyme to assist that enzyme's function. Okay, they get cycled through reactions, and that's where these terms here come from, and they release energy. So that's their main job, is to release energy for reactions that occur within cells. An unloaded coenzyme is a coenzyme that hasn't picked up a chemical group. So that is not able to release energy. It's almost like an uncharged battery. Whereas a loaded coenzyme has picked up or accepted a chemical group, and when it offloads that, it will release energy. Okay, so if we look over here, this is our enzyme, it says E, and this would be our coenzyme. When you see this reaction occurring over here, so we still have a substrate that will bind and the products will be created like we would in a normal substrate enzyme reaction. This was our coenzyme. It started as a loaded coenzyme. This was the coenzyme, and this could have been, for example, hydrogen. Okay, that was the chemical group that it had to pick up. After the reaction, they get split up. So this is, the coenzyme hasn't changed, it just isn't loaded anymore. So now the hydrogen has separated, they haven't been damaged, but this is now an unloaded coenzyme. It can't release any energy until it gets cycled, picks up another hydrogen, and is now a loaded coenzyme again. The examples the coenzymes we need to look at in photosynthesis are NADP, which is unloaded and becomes NADPH, which is loaded, it's picked up a hydrogen, and ADP, which is unloaded, and when it picks up a third phosphate, it becomes ATP and is a loaded coenzyme. Right, this is kind of the whole picture of photosynthesis. I'm just going to draw a little line down here. We have two stages, you do need to know both, the light dependent stage and the light independent stage. Okay, all of the things that we wrote in our equations are here, and then there's a few additional things as well. So if we remember, our inputs for photosynthesis are water and carbon dioxide. So they're both there. Our outputs, so we also have our light input. Our outputs are oxygen and glucose. We'll look at each of these in a bit more detail. Just make note of these additional inputs here, which are not written in our equation, but they're crucial for the process to happen. So our NADP, which is our unloaded enzyme, ADP, which is an unloaded enzyme, and also the phosphate that is going to have to join with that ADP. Okay, so you do need to know the inputs and outputs of each stage. So we'll just look at this left side first, the light dependent stage. Okay, we call this the photo stage because this is when the light is absorbed, and this, as we said before, occurs in the thylakoid, so in those kind of membrane sacs. Inputs: light, water, ADP, inorganic phosphate, and NADP. So from our equation, you probably only recognize these two, the light and the water, but the other things are crucial. These steps. First, the light energy is trapped by the chlorophyll, so it is absorbed, and remember that's red and blue light most. That light energy is used to break those water molecules and separate them into hydrogen and oxygen molecules. Okay, so those bonds are broken and the H2O is separated into H and O. The oxygen at that point binds with another oxygen to become an O2 molecule, and they are disposed then as waste. They're not waste to us, obviously, that's the oxygen that we breathe, and that's really important. But that is our first output of the light dependent stage. That oxygen doesn't stay on for the rest of photosynthesis to occur. The ADP, the unloaded coenzyme, and the phosphate interact with an enzyme called ATP synthase, which its job is just to synthesize ATP. So basically we add the ADP to inorganic phosphate, so aniline diphosphate plus a phosphate equals ATP, so that ATP is produced. And we'll consider that an output because it will move through to the next stage of photosynthesis. We also have a similar thing happening with our unloaded NADP, the coenzymes, picking up the hydrogen atoms that were made up here. So remember, our water got split. The oxygen part of that has been released, but we need to move this hydrogen. Okay, so the NADP pick up the hydrogen atoms to become NADPH. So they are our loaded coenzymes, which you can see over here. And then the NADPH. The NADPH and the ATP leave the thylakoid and will move into the stroma. Okay, so these are important. They're outputs of this stage. As you'll see in the next slide, they are inputs for the light independent stage. Okay, so we had our light dependent stage over here. Now we're moving into our light independent stage. And we can see here those ATP and NADPH that were produced in the first stage carry over here. Okay, this is our synthesis stage, the building stage. It occurs in the stroma, in that liquid. Our inputs, NADPH and ATP from the light dependent stage, and also carbon dioxide. Okay, our processes, the ATP and the hydrogen have been brought across. The hydrogen was ferried on this NADPH. The carbon dioxide molecules diffuse from the atmosphere in this stage. Okay, so they diffuse through those little stroma in the bottom of the leaf. Then we have a process called the Calvin cycle. And this comes up in the exam quite often in kind of a diagram, which you'll get the chance to look at some point. So in the Calvin cycle, one carbon from the carbon dioxide molecule combines with a 5-carbon molecule called RUBP. So we get 5 plus 1 equals a 6-carbon molecule. Okay, that 6-carbon molecule immediately breaks into a 3-carbon molecule and another 3-carbon molecule. One of these compounds will leave the cycle and will go through more changes, but it will eventually become part of our glucose molecule. One of them will stay in that cycle, which is why we call it a Calvin cycle, and will continue to create RUBP. You don't need to know much more detail than that about it, but you do need to know that this cycle occurs, and from this cycle, some carbon gets contributed to our final glucose molecule. The NADPH, which had come from the light dependent stage, lets go of its hydrogen atom, so it drops it off, and that now has returned to being NADP. It's unloaded because that hydrogen is gone. The ATP, remember ATP's main job is to release energy, so the ATP unloads a phosphate. It's now become ADP and phosphate. Remember, they don't get damaged in that process, they just join and unjoin. So that has released energy that will be used to build a glucose molecule from carbon, hydrogen, and oxygen atoms. So remember, this carbon had come from our carbon dioxide. this hydrogen had originally come from our water in the light dependent stage, but has now been carried on our NADP, and the oxygen has also come from that CO2. So they were all broken up. Now they'll get joined to form a glucose molecule, 6 carbons, 12 hydrogens, and 1 oxygen. Okay. It is worth knowing there's also a few leftover hydrogen and oxygen atoms, and they form H2O molecules, water molecules. Okay, so that is an output of the light independent stage. When we write our equations, we balance the equation, so we don't include, there's water that goes in and water that comes out. They balance out, so we don't write them in our equations. We don't have to, but it is worthwhile noting if you were to kind of, use little markers to count up all the atoms that go in and come out, you'd probably find you've got some hydrogens and oxygens left over at the end. They become a few water molecules. Right, this is another way of just showing the inputs and outputs of the light dependent and light independent reactions. If you prefer the diagram, you don't need to use this. If this makes more sense, you can use this. So we have our kind of additional inputs. We have water goes into our light dependent reactions. You might also want to add in there, you have your light coming in. Okay, water goes in, oxygen is released as an output in that stage. NADPH and ATP get carried over into the light independent reactions, so they go into that. We also get our carbon dioxide from the atmosphere, enters the stroma for that, and we have our outputs, glucose, water, and our additional outputs, which you'll notice they come out of the light independent reactions the same way they went into the light dependent reactions. That's that cycle again. Okay, excuse me. If you understand the inputs and outputs of photosynthesis and you know a bit about things like enzyme reactions, you could probably figure out how each of these factors might affect the rate of photosynthesis. Okay, so we'll start with light intensity. We're talking here about if something is going to limit photosynthesis, so make it decrease. So if there is not enough light, basically we don't have the light energy to split the water molecules. And that's one of the first things that occurs. Remember, if you're writing in an exam, you need to be specific. You can't just say there's no light, so there's no energy, or light is a required input for photosynthesis. You need to be specific about what it does. So if there's not enough light energy, that means the water molecules can't be split into hydrogen and oxygen molecules. That would decrease the rate of photosynthesis. Let's say we don't have enough carbon dioxide in the air. That means we don't have enough carbon and oxygen molecules, atoms, to form the glucose molecule. Remember, you need 6 carbons and 6 oxygens for that glucose molecule. So if there's not enough carbon dioxide in the air or in the environment, we need to get them from somewhere, and we don't have them. If the temperature is too low, now remember this is different to light, we're talking purely about temperature here. If the temperature is below the optimal temperature of the enzymes of that plant, you would say that enzyme reactions will be slowed. You can even think back to the topic we did on enzymes, where you could talk about how substrates and enzymes can't collide fast enough to create reactions. And on the other end of that, if the temperature is above the critical temperature, well, if it's above the optimal temperature of the enzyme, the reactions will start to slow as enzymes may damage. And then if we go above that critical temperature, reactions will stop completely as the enzyme will denature. Okay, so remember the structural integrity of that enzyme will be gone, the enzyme would denature, and no reactions would occur. So you'll see with light, we kind of get this nice graph that goes up. Once you reach the perfect amount of light, it often can't really go any higher than that. The rate is occurring as fast as it can. Same as carbon dioxide, you could have heaps of carbon dioxide available, but the plant can only photosynthesize at the rate that it can do it. But with temperature, note that if it's below optimal temperature, it's kind of a lower rate. Here at optimal temperature, it's photosynthesizing at its best rate, and then it might decrease, and then you might even get a more sudden drop off if we hit our critical temperature and the enzymes had denatured. Here, we'll look at this stuff more when we look at the relationships between photosynthesis and cellular respiration, but just note that you can have a kind of arbitrary amount where the rate of photosynthesis and the rate of respiration can be exactly the same, which means that we don't have any net uptake or release of oxygen or carbon dioxide, but we'll look at that more later. This is another diagram. Some people might like this. We've got our two stages. We've got the light dependent stage, the light independent stage. So we start on the left, we have our sunlight going in. We know that our input, our molecular input for the light dependent stage is water, H2O, and we get some oxygen released into the atmosphere. Then, if you move down to down here, the things that are going into the light independent stage, this is our energy. So remember, we have energy in the form of ATP, moves from the light dependent to light independent stages, and we had our NADPH loaded coenzyme. So they will go into the stroma for the light independent stage. This is our Calvin cycle, remember, where the carbon and the RUBP get processed, or get cycled, sorry. So we have to get our carbon dioxide coming in through there, and we will have our final glucose molecule. Okay, that is the whole aim of the game. C6H12O6, and then also the things that get cycled back into the light dependent stages, or back into the thylakoid for those to occur. We have our ADP and our phosphate, because that ATP has released the phosphate, and our NADPH has returned to NADP. Okay, so another way of just showing the same things we've talked about. You could also look at something like this, very similar thing. This will always give you an indication that we're talking about the Calvin cycle, just the way that that diagram is shown. So we would have we know this is our granum, it's a stack of thylakoids. We'd have our water comes in, our oxygen comes out, our ATP and our NADPH travel across, carbon dioxide comes in, glucose goes out, ADP, phosphate, and NADP gets cycled. Okay, the other video looks more at cellular respiration. It's really important that you're always thinking about the relationship between these two very important processes. You'll notice that we looked at photosynthesis today. The inputs of photosynthesis are carbon dioxide and water, are outputs of cellular respiration. And then the outputs of photosynthesis, oxygen and glucose, are inputs for cellular respiration. Okay, so it is a beautiful cycle, and a lot of questions come up in the exam that ask you to look at the relationship between those two. So you should definitely have a diagram like this that you can keep referring back to.